Okay, so this is a very long chapter, chapter 10, sensory systems. So we're going to take 2 class periods to finish this up. This lecture will deal with just the general principles of the sensory physiology and the somatosensory system. So this is a cartoon from the Awkward Yeti. You can look over it. It's really cute. And one of the things that your book starts off with at first is just the difference between sensory and then what's in the real world, and then perception. So the word perception is what your brain does with the information coming into it and how it maybe changes it in your understanding of what's happening in the world. So what your brain is getting all the time is electrical impulses, right? It's in the dark. It can't hear itself. It can't see anything. It can't feel anything. So it relies on electrical impulses coming from different pathways, different neurons. They are going to come in with electrical signals and go to different areas of the brain. And your brain will put together the perception of that sense. So the brain is working all the time with it's getting bombarded with all different kinds of electrical impulses. And it has to be able to make sense of those impulses in a way that helps you survive and navigate the world. And sometimes that means changing what is actually out there to make it more for the perception to make more sense to you, because your brain is also using all that it's learned in the history, the context of what the senses are coming, like what's the context, what does the world look like, even your mood, and the people around you, or what you're doing. All these things can change your perception of a certain sensory input. So your book begins with these examples of, you're looking at a picture of 4 green semicircles, like a little Pac-Man. But what you probably are really seeing is a white square, right? A white square that's separate from the white field in the background. And this is because your brain is kind of inventing that square, right? There's no line connecting these edges, but your brain has done it for you. So you've completed the square. And this is because this is the difference between perception, right? There's actually no square there, but your brain is perceiving it. Because it's basically, connecting a few dots for you very, very quickly, so that, perhaps this helped you in some way in nature in the past. This is another example of where you have these concentric circles, and it can modify the square. So the square is actually a straight square, but perhaps you see the walls of the square a little bit curved. Again, this is your brain changing what's actually coming into your eyes, right? And the perception might be different than the reality. And this is another example of that. So your brain doesn't know if this is a duck or if it's a bunny with ears off to the back of its head. And because of this, it can flip-flop between the two depending on where your eye falls, right? It's equally likely that it's a duck or a bunny. And so your brain just kind of flips between the two. So your perception of the same image can have different meanings based on what your brain wants to perceive at that moment. Okay, so let's start with the physiology, receptor physiology. And just so you know that receptors of your body, right, the way that we detect anything in the world is through receptors. And if we don't have a receptor for it, we don't feel it. So there are many things that we don't perceive. We don't perceive radio waves. We don't perceive other colors of the rainbow. Some animals can see ultraviolet. We have hearing that's only between a certain frequency set. So what we have evolved to sense in the world is what we needed as human beings to survive. And other animals and other organisms, even plants, they've evolved other detection systems with their specialized receptors to detect what they need to survive in the world. So these are some receptor endings. So some of our receptors are actually just the afferent neuron themselves. So the afferent neuron has a specialized ending. And so you can see the stimulus comes in one end. Here's the nerve ending in yellow, and it's going to create an action potential. So the stimulus comes in, it's going to change the ion permeability, right, in the nerve ending. So whatever the stimulus is, it could be pressure, or it could be, heat, or it could be something like that, opens or closes some ion channels, creating the receptor potential, and then eventually creating an action potential that goes to the central nervous system. So remember the afferent. A means sensory, so we're moving towards the central nervous system. Also, we have specialized cells. So this is going to be a separate cell, right? A separate cell from the afferent neuron, and it's going to act as our sensory receptor. It's going to communicate with the afferent neuron. Okay, so the stimulus actually comes and stimulates the cell here. We can see that the cell contains a voltage-gated calcium channel. In this example, calcium channels open. They're going to create, the calcium channels will help exocytosis, right? The neurotransmitters will be packaged up and moved to the plasma membrane, released, and then you have the receptor on the afferent neuron end, and then an action potential can be sent to the nervous system. So either of these two scenarios, right? Either the nerve ending itself acts as the receptor, or we have a specialized cell that acts as a receptor. So the stimulus is going to act on this other cell, and that cell communicates with the afferent neuron, which then brings it into the central nervous system. All right, so let's talk about adaptation. So your senses, some of them can adapt, meaning that you're going to have a decrease in the amplitude of the receptor potential over time, and it's going to decrease the amount of action potentials that you're creating. Okay, so what we're going to look at is this graph. So both of these pink graphs down below are showing you the same stimulus. There's a stimulus on, the stimulus comes on, the stimulus does not change over the course of the time span, and then the stimulus comes off, right? So it's the same on either side. If you have a slowly adapting receptor, slowly means that the receptors will keep working even with the prolonged stimulus. So we see a nice big receptor potential at the initiation of the stimulus, and then it does wane, right? It does go down. You don't have as much of a receptor potential anymore, but and then when it turns off, it goes away. But if you look at the rapidly adapting receptors, you have a big peak of receptor potential at the onset of the stimulus, and then it just goes right back to resting, right? So here's resting potential. It just goes right back to resting potential. And then there's a curious little hyperpolarization that happens when the stimulus comes off. So what this means is a slowly adapting, also called a tonic receptor, it will keep working with prolonged stimulus. So you will constantly get the perception of the stimulus still being there. So muscle stretch, skin pressure, those are both considered slowly adapting. Rapidly adapting means that you're very quickly going to lessen the perception of that stimulus, right? So smell, olfaction works like this. If you walk into a room, let's say you walk into a pizzeria, initially the intense smell of pizza is, overwhelming, and everybody, makes a comment, but a few minutes later, or even a minute later, that smell goes away, right? It's no longer as intense, and then just becomes background. So that's what your rapidly adapting receptors will do. They give that intense signal at first, but then they basically shut off. So pressure in your skin is the same way. And so the reason why we have slowly and rapidly adapting receptors is because your brain is getting information all the time from all different parts of your body, and it has to, it can't get overloaded, right? So there's ways that your body has figured out to not overload your brain with stimulation, and that way the brain can concentrate on what's most important for you to survive in the world. Okay, so through evolution, your body has modified these nerve endings so that you pay attention, or what's basically more important for survival, maybe keeping pain away, is going to be more slowly adapting, right? To constantly give you that information that something's happening to your body, whereas maybe things that are not as threatening will be rapidly adapting. All right, So let's look at our pathways. This is a generalized pathway for sensory systems. So when you have a pathway, you usually have several neurons that are in that pathway going into the central nervous system. So we call them first order, second order, and third order neurons. Your first order neuron is usually the afferent neuron. So the stimulus will come in. Whether you have a separate cell here or not, it doesn't matter. It's the neurons we're looking at. So we have stimulation of the receptor. The first order neuron, will synapse with the central nervous system. And in the central nervous system, we will have a second order neuron. So most of the time, if you're talking about touch, this will be, pain and temperature, for example. The first order neuron is the sensory neuron that's in your skin, and then that will travel to the spinal cord where you find the second order neuron. So it'll synapse with a neuron in the spinal cord, which is the green one, second order neuron. Then that will travel to the brain. And for sensory systems, this is going to be, remember the in anatomy, the thalamus of the brain is the relay station for all senses. So your second order neuron will synapse with the thalamus, and in the thalamus you have your third order neuron, which brings it to your specialized region in the cortex for interpretation. So if this is touch, it might go to the somatosensory cortex in the parietal lobe, right? If this is hearing, it'll go to your temporal lobe. If this is vision, it will go to your occipital lobe. So that third order neuron will bring it to the cortex. Now this is just general. Some pathways only have two neurons, but this is in general, and I want to show you an example of this. So this is just a review of the different cortices in your brain, right? The somatosensory cortex is where all the touch, right? Touch and stretch, things from your skin, your joints, your bones are going to come, and muscles are going to go to the somatosensory cortex. Then gustatory cortex is for taste. That was in your insula. The visual cortex is where you interpret vision. That's in your occipital lobe. The olfactory cortex is also part of your temporal lobe. Auditory cortex is temporal lobe. Right, vestibular cortex also in a little bit of the parietal lobe and temporal lobe. Anyhow, looking at the spinothalamic tract. So this is an example of how pain and temperature are traveling to the brain. So you're going to have your first order neurons here. They're your afferent neurons, right? So this is going to be in your skin somewhere, some nociceptors or thermoreceptors. These are going to travel into the spinal cord. You should remember this from anatomy, that your afferent neuron, your sensory neuron, comes into the posterior side. This is your dorsal root ganglion. And then your soma of that sensory neuron is there in the dorsal root ganglion. It's going to synapse with an interneuron in the spinal cord. So this is your second order neuron here. Okay, the second order neuron was in the spinal cord. The spinal cord, the second order neuron will go up through the brainstem, enter to the thalamus, right? We have another synapse in the thalamus, and then that third order neuron is going to bring that action potential into the primary somatosensory cortex, because we're talking about pain and temperature. This is going to be interpreted here at the primary somatosensory cortex of the parietal lobe. So that's basically how that information would flow from the peripheral nervous system to the central nervous system for interpretation. All right, so our third letter C, sensory coding. So how does the brain know what the stimulus is, how intense it is, where it is? So first of all, the number one is basically the type of stimulus has its own specific pathway. So coming back to this picture, pain and temperature are going to go through this pathway, and every sense that you have on the body is going to have its own specific pathway, its own set of neurons as it reaches the brain. And the brain will know where it comes from in the body by what neurons it takes. So the stimulus type was going to it's going to correspond to the pathway, right? There's a specific pathway to every specific stimulus. Now for intensity, that is where we look at this. Intensity is going to be coded by the frequency of action potentials. How many action potentials are created per unit time? So in this example, it's looking at the action potentials here, right? So let's take a look at our graphs. So here's our stimulus strength. So let's look at our first one. The stimulus comes on, the stimulus goes off, and here's our receptor potential. So the onset, we have our first receptor potential, and then we have some action potentials being generated. We can see that it's kind of going down, right? The receptor potential is starting to adapt a little bit. And with that downward slope, we actually see that the action potentials are getting more and more spaced apart, right? So as we decrease the intensity that's generated by adaptation, we're seeing that the action potentials are further apart. It's more intense, right, at the beginning, where it corresponds to a more intense receptor potential. Now let's look at a bigger stimulus, right? So this graph is stimulus strength. We have a larger stimulus now. We have a larger receptor potential. And look what happens in terms of our action potentials. We have much more frequent action potentials, right? So one right after the other, even though the time is the same. So the time span, both of these are the same, but we have a lot more action potentials over that time span. And we can also see that same pattern happening with adaptation, right? The graph is going down a little bit. We can see that at the point where it's at the end here, the action potentials are a little bit more spaced out. So the intensity of the signals coming in is coded by the frequency of the action potentials. The more frequent the action potentials, the more intense the signal is going to be interpreted in the brain. Another thing is the location. Let's talk about the number of receptors. And I don't know if I actually have a picture of that. All right, I don't have a picture of this one. It's in your textbook, where the number of receptors being activated also can dictate a larger stimulus. So if there's one neuron coming down and the terminal arborizations are split, right? And we have a very small stimulus, then it's only going to activate a few receptors on that one portion. But if we have a greater stimulus, right, then it's going to activate more receptors of that same neuron. You can also, so this is the same neuron, but you can also activate and recruit other neurons. So the neurons are going to overlap a little bit in their receptive fields. And if you have a few neurons that are very close together and the stimulus is here, it might, if it's a small stimulus, it might only stimulate one neuron, but if it's greater stimulus, it can actually stimulate two or another neuron over here. So it can recruit more and more neurons the greater the stimulus is, which will then get coded as a greater stimulus in the brain. And our last topic here is just for stimulus location. So how can you locate something on the skin? So every neuron is going to have a receptive field. Well, not every, I'll say that. The somatosensory part, right? Touch and pain and temperature and things like that. They will have their receptive fields. So a receptive field is just the area of your body that one afferent neuron is going to monitor. And sometimes you can see the receptive fields can overlap. But these can be different neurons. So this can be a, deep pressure. This one can be pain. So they can overlap a little bit. But in terms of touch, right, there is something called the, lateral. This is lateral inhibition. Let's actually just talk about receptive fields first. So this is a receptive field. Okay, so if you were to stimulate one spot on this receptive field here, within the receptive field, it's going to feel as one point, right? So if you touch anywhere within this receptive field where my X is, that's going to be 1 point. If I have two close together points, like if I have two toothpicks and I have stimulated just one receptive field, but actually I have 2 points, I'm still going to perceive one point because I've only activated one receptive field. So when there is a large receptive field, that means that it's going to be harder to locate 2 points, right? It's going to be harder to really locate and discern or discriminate, I should put discriminate, right, between 2 points. However, sorry, my writing is very messy. However, the smaller the receptive fields you have, right, then if I have my X here and another X here, I can actually hit 2 receptive fields, and then I'm going to feel 2 separate points. So when you have smaller receptive fields in the body, you're going to be able to be more discriminative in basically pinpointing the exact location of a stimulus. So receptive fields are going to vary on your body. You can see that this is a little table of different receptive fields. So this is that 2 points discrimination threshold. Your lips have the greatest acuity. Each receptive field for touch there is about 1 millimeter, right? So very small area. So that means that you can, if you have 2 points at 1 millimeter apart, you'll probably feel 2 points. Your index finger is also very, very sensitive. Your palmed hands, where you get less and less acute, might be your calf, right? Your calves are not really built as parts of your body that are going to be used for fine acuity, right? For feeling yourself around the world. That's just not what your calves are for. So you don't really need very small receptive fields there. You can have very, very large receptive fields there. So your back, so these larger areas of your body that really are not built for dexterity or navigating the small objects in the world, they're not going to have small receptive fields. They're going to have very large receptive fields. All right, so let's go look at some receptors, but just want to talk about lateral inhibition for a second. So lateral inhibition just means that a stimulus can actually be inhibited by other neurons after the stimulus has taken place. So let me see if I can make sense of this. So here are some afferent neurons. Here's X1, Y1, and Z1. We can see that Y1 is firing the most action potentials. And X1 and Z1 are also firing action potentials. So the stimulus is going to encompass most intensely at Y1, less intensely at your other two. But so think about this as being a pencil tip on your finger, right? So here's your finger and here's your pencil tip. And you can actually feel the point exactly where the pencil tip hits, even though the skin on either side of the pencil point is also being pushed down on. Like there's some deformation happening on either side. Your brain then, so if all 3 of these receptors were being stimulated, then your brain would think that you have a very large pencil tip hitting that entire area of all 3 neurons being stimulated. But in fact, it's not, because we have this lateral inhibition. We have some inhibitory neurons on either side here that can actually dampen, you can see what's happening here, dampens the action potential coming from the other neurons on either side of the great stimulus, and allowing the just that one point to have with the greatest action potential. So that way in your brain, it can be interpreted as this point is very intense and there's almost nothing on either side, rather than sort of 3 moderate, 1 intense but 2 moderate, right? We're just going to inhibit the lateral neurons so that we can allow that the 1 intensely firing neuron to bring the information to the brain, so you can have these finer discrimination points on the body. Okay, so let's take a look at our next topic here, just with some different kinds of senses. So we're going to look at mechanoreceptors in the skin, and thermoreceptors and nociceptors, and some pain perception. So these are some somatosensory receptors. Proprioceptors are position of the body, so this is going to be in your joints your joint capsules, in your tendons, and your ligaments. So you can feel the position of your joints, you can feel the stretch of your muscles. And so you would know the position of your body in space. So this is your body in space, right? You can close your eyes and you can know if your legs are straight or bent by this. Proprioceptors. Mechanoreceptors are going to be receptors that are stimulated by some kind of physical deformation, so some kind of physical bending or pushing or smushing or some kind of physical deformation. Thermoreceptors are going to be stimulated as they sound by temperature, different temperatures. And the nociceptors are stimulated by, nociceptors can be stimulated by many stimuli, right? But it ends up as being interpreted as pain. So when you stimulate a nociceptor, you feel pain. What can cause pain is different, right? So we have some nociceptors, we'll look at this in just a second, but some nociceptors respond to chemicals, some nociceptors respond to physical deformation. Oops, sorry. All right, so these are the sort of a generalization or table of different receptors in the skin. So here are and this is just skin, right? So mechanoreceptors, these are deformed, right? So if we take a look at the mechanoreceptors here, we can see that they have different fiber types. And this, I didn't stress this in the past, and you don't need to worry about it right now, but the different sizes of the axons and are going to deliver the message faster or slower, right? Remember, larger diameter axons are going to deliver the messages very quickly. Smaller diameter axons will measure it more or carry the information more slowly. Anyways, but this is what that is looking at. So we can see that we have the locations. And so we just want to just stick to what's in your outline. Piscinian corpuscles are deeper pressure. Let's just actually take a look at it. So piscinian corpuscles are deeper pressure. They are deeper in the dermis. So they're going to be mechano. So you have to push down hard enough on the skin where it actually reaches this bottom layer of the dermis to deform the connective tissue that you see wrapped around the nerve ending. So the nerve ending comes in like this, and it's right here in the middle. There's connective tissue all around, and so you have to push your way through that connective tissue in order to hit or deform that nerve ending. So that's why it's going to sense deeper pressure, right? Because it's you do require some pressure to stimulate that corpuscle. Meissner's corpuscles are going to be more superficial, right? So you can see Meissner's corpuscles are really next to the epidermis, where you have finer texture and fine detailed for touch. And these are only going to be found in the hairless areas of your body, which is actually called glabrous skin. So if you see on the table glabrous skin, that's hairless. So the palms of your hands and the soles of your feet will have Meissner's corpuscles. And the reason why people can read Braille, right, is because of these Meissner's corpuscles. They're very, very good at detecting texture and pattern. Hair follicle receptors are going to detect the movement of hair. And then for slowly adapting receptors, we have some free nerve endings. We have, and free nerve endings are pain and temperature. So here's our free nerve ending. We can see just the nerve endings are found in the epidermis, and they are going to be very, very sensitive to pain and temperature. And then we have Merkel discs, which are also very high up in this in the epidermis, and this for very, very light touch. And then we have Ruffini's endings or Ruffini's corpuscles, which are deeper down the dermis again, and they're going to be for deeper, lower vibration. Okay, so we have a lot of different kinds of touch receptors in the skin, and just looking at the difference here with hairy skin and glabrous skin or hairless skin. All right, looking at our next topic, it's going to be thermoreceptors. So the thermoreceptors in the skin, The temperature is actually going to open and close these ion channels. So they're temperature sensitive ion channels, which is also called transient receptor potential, which is also relates to transient receptor potential. So temperatures will create a transient receptor potential. And you can see that we have warm and cold. So we have two kinds, right? Warm receptors and cold receptors. These are going to respond to just different ranges of temperature. There's no need to memorize this. But the intensity, right, increased frequency will increase in temperature. So as you're moving into the higher temperatures of the temperature range that these warm receptors are detecting, you're going to get more action potentials. And the opposite is for cold. There's an increased frequency with a decrease in temperature. So as you move, it's the same direction. As you move colder, you're going to get more and more action potentials. What's interesting is that these thermoreceptors can also respond to different chemicals. So warm, right, can also respond to camphor, capsaicin, which is the hot spiciness and hot peppers. So when you eat a very spicy pepper, like a habanero pepper, you can actually feel heat, but that's because the thermoreceptors are responding to the capsaicin. And then the ethanol can also give you the sense of heat, right? Because if you have a shot of alcohol, you can feel the warmth of that alcohol. One thing that's kind of interesting too is that cold receptors, you can see that this is the graph of cold receptors. So here's our cold receptor span. Here's our warm receptor temperature span. And then look what cold does again. So above 45 degrees Celsius, which is very, very warm, your cold receptors will go up. Again, they'll get activated by very, very warm. And so you're interpreting this hot temperature as cold because it is the cold receptor that's sending that signal to the brain. So the perception is cold even though the actual temperature is very hot. So that's called paradoxical cold. All right, and then cold, by the way, also is stimulated by menthol, which gives you a cooling sensation, and eucalyptus oil. Okay, so nociceptors. Nociceptors, again, they're going to be free nerve endings, and they're going to respond to a variety of stimuli because they are going to deliver the pain perception, but different things can cause pain. So I want to just show you this table here where we have nociceptors, and that capsaicin, the same thing that activates warm receptors, right, that hot pepper, can also activate pain. So if you're in pain because you ate a hot pepper, that's very true, right? You're stimulating your nociceptors. H plus is acid. So very strong acid can also elicit pain, and camphor can also elicit pain. So for nociceptors, they're going to be responding to noxious stimuli. Noxious means anything that's potentially tissue damaging. And so sometimes when your tissue is damaged, the chemicals that are being sort of released from damaged tissue will activate nociceptors, and this is a really good thing. Pain is actually very, very helpful and beneficial sense to have because it tells you to stop doing something that's potentially harmful to your body. And there's lots of different kinds of factors that go into your sensation of pain. So before we look at this picture, I just want to mention that, there are nociceptors that respond to mechanical stress, like if you stub your toe, right, or you twist your arm or ankle. Thermal is intense heat, and then polymodal nociceptors are sort of able to respond to many different kinds of stimuli, chemicals as well as temperature as well as mechanical stimuli. Okay, so looking at the pain response, right? So we have pain here. This is our pain response in the middle. Your response to pain is it's based on a lot of different factors, and some of those factors are what's coming in directly from the senses, but can also be what mood you're in. Are you anxious? Are you depressed? Are you feeling good? It turns out that if you are sad or depressed, you're going to be more likely to perceive more pain, right? So if you're sick, for example, people who are sick, if you're ill, you're going to be more readily, you're going to perceive pain with a lower stimulus. So physiological, right? So this is, what's coming in from the nervous system, the location, the onset, the duration. Etiology is what's causing it. But what's interesting is also the cognitive, right? So you can sort of think about other factors that go into pain, especially if you deal with chronic pain. There are coping mechanisms that you can use. Your attention to pain, right? So if you pay attention to the pain, it's going to be overwhelming. But if you move your attention away from the pain, it actually decreases the perception. So there's actually really interesting virtual reality goggles that help people with chronic pain take their mind away from that. There's behavioral. What are you doing physically? Like imagine you're a soldier, for example, who gets severely injured, might not actually feel pain just because there's so much else going on around the soldier that the actual perception of pain is very, very minimal. And not until after the soldier is taken away, maybe to a hospital, maybe an hour later when he gets to calm down, then the pain starts to come in, right? So there's this, what you're doing physically, what are your, all these different factors. There's interpersonal interactions, communication, what medications you're on. And then even here, socially and culturally, some people are just going to be brought up with different perceptions on what pain should be or who is allowed to feel pain and how much pain are you allowed to show. And that might actually mediate your perception of pain. So pain is a very, very interesting, multidimensional feeling, right? So that's just what I have in the pain response. For referred pain, this is going to be for visceral pain. So it's interesting because your viscera have pain receptors, right? So we can feel our organs if we're not feeling good interiorly, like our stomach hurts, our intestines are hurting. You have a heart attack, right? Those afferent sensory signals coming from your viscera are going to come in, with those on the somatic side. This is somatic body. And then they're going to synapse with the same secondary neuron, right? Second order neuron. So what happens in referred pain is that even though the heart is the organ that's in pain, you will oftentimes feel pain in the somatic part, meaning the skin, the muscle, right? Or even bone sometimes. But the skin is really interesting for the heart, right? So heart attacks, you'll oftentimes feel numbness down the arm, right? Or some kind of pain here. Sometimes people get jaw pain. Sometimes there's back pain. But this is because throughout your entire life, you've only known somatic pain, right? A child really doesn't know a heart attack pain. So the brain doesn't learn to interpret visceral pain separately than somatic pain. So when you do encounter visceral pain, it gets mixed up with that somatic pain. So you have both. You're going to have the visceral pain and then you also have the somatic pain because your brain thinks that it's also, because it's coming from sort of the same secondary neuron, that it's also going to encompass that somatic part. Anyway, so this is called referred pain. Referred pain is pain that stems from the viscera. It stems from an organ, but it's actually felt on the somatic parts of your body. It's referred to your upper parts of your body. And so for modulation of pain signals, right? So there can be different ways to treat pain. The most common way to treat pain is with drugs that can block it can stimulate the opioid system, so things like morphine. And that's actually something what letter B, the brain can produce endogenous analgesia to dampen the perception of pain. So your body can actually make endogenous analgesics. These are going to be your opioids. So the enkephalin here, you can see. So if you have a source of pain, we can see that we have this endogenous opioid being made here, right? And that can dampen the messages coming into the brain. And also the brain itself can create some endogenous opioids. And this is how drugs work, right? Like morphine, some very, very powerful opioid drugs. They work to dampen the perception of pain by inhibiting those pathways where that pain takes. Opioids being very, very dangerous because they're very addictive. But the second thing is that non-painful mechanical stimuli can sometimes block the pain pathway. So if you hurt yourself, if you stub your toe, or if you hit your shin on the coffee table, sometimes you go and you hold it and you lightly rub that area, right? So that light rubbing is a non-painful mechanical stimulus, and they think that that can actually block the pain pathway. So the two kind of work against one another. And so because you're feeling that non-painful stimulus, it dampens down the amount of pain that can get to your brain. So it's relieve pain that way. And there's TENS therapy. TENS is this therapy for people with chronic pain. It's basically just delivering a vibration into their body, and then that will serve to block the pain pathways that someone is experiencing. Okay, so that is it for our first part of the lecture.